So the first study in this group is actually the second year of funding for Dr. Tay, who is at Mass General, and he has been developing iPS cells or stem cells that have deletions of like the big deletion in PWS, the smaller deletion in PWS, and then individual genes. And he's done all of this on the same genetic background, so the same cell line using CRISPR technology. And that allows him to do a really robust comparison of what happens when you're missing SNORD116, what happens when you're just missing MAGEL2, what happens when you're missing all of them. So he has been working in the Talkowski lab, and that lab does a lot of this kind of work for all kinds of neurodevelopmental disorders, which is actually great because then you can compare PWS to other neurodevelopmental disorders. And the goal here is to characterize the cellular properties of these PWS stem cells after they are differentiated or encouraged to become like hypothalamic neurons in a dish. So they essentially take the stem cells, they feed them things to make them essentially turn into brain cells, and then they can study a PWS brain in a dish. So why are we excited about this study? This lab has some of the most advanced technologies for looking at overall global changes when you disrupt specific genes. And they have a pipeline that they send these cells through again, and they can compare to other neurodevelopmental disorders to see what's the same and different. And that's important because maybe there's some shared pathways and shared therapeutic avenues that we can pursue. This is a powerful resource for the entire PWS research community. So this lab has been great about sharing it with other researchers so they can do their studies. And ultimately, it may support the development of high throughput screening approaches. So if you define differences in the biochemistry or the electrical interaction of these neurons, then you may be able to screen drugs and see what restores it back to a more normal level. So the long-term goal of this study is to provide new cellular models for PWS to understand the changes that happen when we are missing those PWS genes and identify pathways, druggable pathways for new therapies.